The overall goal of this procedure is to maximize the information of the molecular readouts associated with the regulation of dopamine from a single tissue dissection from the somatodendritic regions of the midbrain. This is accomplished by first making a precise dissection to segregate the Substantia Nigra from the ventral tegmental area. The second step is to process the tissues for the analysis of dopamine by HPLC and further processing of the precipitated endogenous protein for analysis. Next, the processing of the precipitated protein for total protein content allows the determination of which dopamine-regulating proteins, or protein phosphorylation, can be measured in conjunction with the readout of dopamine tissue content in the sample. Ultimately, the analyses of dopamine and its regulating proteins in the discrete midbrain somatodendritic regions enable conclusions to be drawn regarding the molecular mechanisms associated with dopamine regulation.
The monoamines affect many behavioral outcomes, and indeed, researchers in the field of addiction biology and locomotor dysfunction, such as notably in Parkinson's disease, are very interested in how monoamines, like dopamine, which we study in my lab, impact behavioral outcomes. But the importance of studying the monoamines can be further embellished if we understand how they are regulated on a molecular level. The benefit of our technique is that we can take from a single sample obtained from CNS tissue multiple readouts from that one sample, so we can get a full readout of how molecular events affect the dysregulation of monoamines. We can take that data and then